Good morning, Mr. Danner and my beloved classmate. Today, Group 2 will be presented about epidemiology. For that, let's meet our group members. First, there is Ahmad Fadoli. Second is Farhan, Anka, Alrafif. Third is me, Randita Kusuma. Fourth, Nurul Handayani. 5, Stevania Nersiartista. 6, Amanda Kurniawati. And the last but not least, there is Tuqya Risang Ayu.
Preliminary. Pathogens are living organisms that are mostly micro and can cause disease in plants. These microorganisms include fungi, bacteria, virus, mycoplasma, nematodes, spiroplasma, and rickettsia. Then, epidemiology is the study of plant disease in population. Epidemic or epiphytotic is a disease that spread from one plant population to another plant population that is wider and in a short period of time. The definition of epidemic is used to show disease dynamic in plant population without considering the severity. Epidemics occur over a period of time or don't always occur all the time. Epidemics occur in certain places, spaces, regions, or are not evenly distributed in every place. This epidemic involves elements of susceptible host plants, virulent pathogens, favorable and environmental conditions, elements of human activity, and element of time. Next. Endemic. A disease that is evenly distributed occurs continuously in every season and originates from the area concerned. Pandemic, a disease that is evenly distributed throughout the continent or the world. Sporadic, a disease is only present here and there with an indefinite interval and doesn't increase.
Definition of epidemiology. The word epidemics comes from Greek, composed of two basic words, namely epos, which means between, one, or above, and demos, which means people, many, or population. Definition of ethnology. Epidemiology of plant diseases means the study of diseases that develop in plant population or the study of plant diseases that possibly develop into epidemics. According to van der Planck, epidemiology is the study of disease in a population. Next. Spread of pathogens. The number 1 is Inoculum Shape. The number 2 is liberation of the inoculum from its substrate. And the last is inoculum holding site.
First is inoculum shape, the part of the pathogen or pathogen that is carried by certain agents that come into contact with plants is called the inoculum or infectious agent. First is fungi. The inoculum can be mycelium, spore, or sclerosium, the inoculum. Second is bacteria, mycoplasma, and viruses. The inoculum consists of individual mycoplasmas and the virus particle themselves. Third is nematode. The inoculum can be eggs, larvae, or adult nematodes. Next is the release of the inoculum from the substrate. Pathogen inoculum has different resistance. Plant pathogenic bacteria do not form spores, but some of them can survive long enough in plant parts, plant debris, and in soil.
Okay, inoculum holding site. First is seed. In the seed through the intuition or cotyledon, namely the seed tissues or embryo, the inoculum survives and is carried on infected seeds or plants, which they grow somewhere or are carried and spread. Second is plant residues, soil, and compost. Plant residues, soil, and compost can be placed to survive inoculum because, in general, fungi can survive in the form of hyphae that are resistant to plant residues, large stems, or small pieces. Fungi or fungal hyphae can survive and adapt to the soil because the environment is moist. There are nutrients from that plants. If you know, viruses usually come from plant residues, and the virus cannot survive without the host in the soil. Plant residues that still have nutrients in them can become a temporary place for pathogens to live. That is different from nematodes. The nematodes can also stay alive in the plant debris, compost, and soil, such as Longidorus spp and Cipinema spp, and transmit to polyhedral nepovirus. Diseases by soil-borne pathogenic fungi are difficult to control because pathogens can survive a long time ago at the host plant, sometimes even having a broad host include weeds. Next. The types of the pathogen spread. Active development and passive development. Next. Active deployment. The water gun mechanism is a mechanism that often occurs in Ascomycetes class mushrooms, whose ascus will swell before maturity and burst, then open the TN, which directs the spores upward with high pressure until the spores are thrown up to height of 100 meters. And second, the cutting mechanism is that the spore cell ends are cut off and the spore can be released into the air. This cutting mechanism is usually found in some Phycomycetes fungi. And the last, the mechanism for releasing basidiospore is that the basidiospores are in a symmetrical position at the end of the sterigma, and the ripe spores will drip liquid from the hilum, and immediately the spores will be released. Next. Passive deployment. First is wind. The spread of pathogens by wind in airborne, such as spores of pathogenic fungi and leaves, stalks, and fruit. And second is water. The spread of pathogens by water is waterborne, caused by the presence of the water droplets or rain, and on the bacterial exudate, causing the bacterials to disperse and spread to various places of contact or penetration. And third, sand, seed, and other plants. Pathogens or inoculum or fungal spores or bacterial cells are often carried in infected seeds and or plant shoots, so they can spread disease to other areas or to other plants. And insect play an important role in this transmission, spread, reproduction, and defense of pathogens. And the last is humans. Humans are long-distance spreaders of plant disease pathogens through the introduction of seeds or shots of infected plants between countries or regions. Next.
Occurrence of an epidemic. The development of the epidemic disease, occurs due to the timely interaction of the elements resulting in the occurrence of plant disease. The elements in question are susceptible host plants, virulent malignant pathogens, favorable environmental conditions for interaction, human intervention, and interaction. In natural system, only 3 elements are considered in interactions that cause disease, namely susceptible host plants, virulent malignant pathogens, environmental conditions that are favorable for interaction, called the disease triangle concept. Next. factor affecting the occurrence of an epidemic. The first is the influence of pathogens on the development of disease epidemics. A. Virulence level. More virulent pathogens will be able to quickly infect their host and produce more inoculum when compared to less virulent pathogens. B. Number and type of inoculum approaching the host. At the same virulence level, the greater the number of propagules found on or near the host plant, the more inoculum that can reach the host, so the greater the chance of an epidemic. C. Pathogen reproduction type. There is a monocyclic, which is one life cycle in 1 year. There is a polycyclic, which is a cycle several times in 1 year. And there is the polyetics of taking several years for a cycle. And D is pathogenic ecology. And E is pathogen spreads. Can cause sudden and large-scale epidemics. Epidemics occur seasonally and locally, such as inoculum emitted by rainstorms. And the ability to cause epidemics depends on contact with plants in the following season. And the second is the influence of plants on the development of disease epidemics. A is level of plant genetic resistance. Host plants that have a higher vertical genetic resistance will not provide an opportunity for pathogens to settle down, so that epidemic disease cannot develop unless there is a new race of pathogens that can attack plant resistance. Host plants that have horizontal genetic resistance might get sick, but the rate of disease development and epidemics will depend on the level of resistance and environmental conditions. B. The degree of genetic uniformity of plants. If the host plant is genetically uniform, planted in a large enough area, the chance of developing new race pathogen will be greater. C. Plant type. In annual crops, epidemics usually develop much faster than in annual crops. D. Plant age. Some plants are resistant when they are very young, when they are old, and before they experience ripening. And the third is the influence of environmental factors on disease epidemics. A. Humidity. Increased humidity can increase tissue succulence, increase fungal sporulation and bacterial division, and helps release spores of several types of fungi. B is temperature. Temperature affects plant resistance, the amount of pathogen inoculum, and affects the reproduction germinations of pathogens. And the last is factors of human activity in these epidemics. A is preparation and selection of planting sites. B, selection of culture material. C, how to cultivate plants. D, how to control disease. And the last is introduction of new pathogens. Next. Epidemic forecast. Some of the requirements needed in forecasting an epidemic. Among others is the one, it is an important plant. For example, food crops, plantation crops which have high value. Number 2 is disease can cause great harm, but only in certain circumstances. Number 3 is there needs to be sufficient information, but observational and research results regarding the influence of various environmental factors on disease development. 4. The growers farmers are required prepare and understand the forecast of disease epidemics. 5 is for the disease in question, there is an appropriate way of controlling it. And the 6 is there is a sufficient time gap between the announcement of the forecast result and the onset of a disease epidemic. Next.
And then epidemiology formula. This is the formula. There are two formula, namely one simple interest disease or monocyclic. Number 2, compound interest disease or polycyclic. With the description, with description. X0 is initial inoculum or number of early disease plant. XT is final inoculum or number of terminally ill plants. E is constant. R is reaction rate or number of sick plant per unit time. T is duration of the epidemic.
Okay, that's the end of our presentation. Thank you for your attention. If you guys have a question about this topic, you all can go to discussion forum in Spada, then write down your question on Q&A with group 3. Thank you.